Hello, I'm Stephen Harrison. I'm going to talk today about classifying sequence variants using the 2015 ACMG AMP guidelines. And this topic is important because differences in variant interpretation can exist. Two equally qualified people could look at the same variant and come to a different conclusion. It could be because of differences in evidence that they're looking at, but also just in the way that they subjectively weight different pieces of evidence. So looking at current data in ClinVar, there's over 164,000 variants that have at least two submitters. Of those, 75% are concordant, with about 22% having differences between VUS or benign and likely benign, and then only 3% being medically significant differences. So overall, laboratories are consistent with regards to classifying variants. However, there still can be differences that occur between any two organizations interpreting variants. So to help with this and to establish more of a consistent framework, in 2015, the American College of Medical Genetics and Genomics, in association with molecular pathology, published this joint guideline in which they go through 28 different evidence types and give each of those evidence types a specific weight. All the evidence types are divided into whether or not they contribute to a pathogenic interpretation, those seen on the right, versus those that contribute to a benign interpretation seen on the left. And then additionally, the different evidence types are weighted within those categories of whether they are supporting, moderate, strong, or a very strong piece of evidence. And so the way that we approach interpretation is as you go through and identify which of these lines of evidence are met by the variant you're assessing. In the end, you're able to take all the different lines of evidence, look at the weight of each one, and then sum those together to reach a classification seen here in this table on the right. So I have an example variant to go through in case you're not familiar with the way that this process works. So I have here a nonsense variant in the gene PAH, and PAH is known to be causal for PKU. So the way I would start is looking at first seeing that this variant is a loss of function variant, and I know that this occurs in a gene where loss of function is a mechanism for disease. So in this case, I'm going to apply the pathogenic very strong code PVS1 for a null variant in a gene where loss of function causes disease. Next, I'm going to see that I can see that this variant was found in only one of about 16,000 alleles in Nomad. And so this is rare enough that I feel comfortable saying that it is absent or almost rare in population databases. And so I will apply PM2, a pathogenic moderate code. And let's say that I found this variant in a case that had PKU, and this person carried a second pathogenic variant in trans. And so I'm going to apply the PM3 rule, another pathogenic moderate code, for this observation. So in total, I have PVS1, PM2, and PM3, so one very strong and two moderate pieces of evidence. If I look then at that chart, I can find that combination of one very strong and two moderates, and see that that reaches a pathogenic interpretation. Overall, moving to the ACMG AMP guidelines has helped laboratories be more consistent in their interpretations. In this study where we had clinical labs reassess variants that previously had differing interpretations, we saw that just using the ACMG framework resolved about a third of these differences. However, inconsistencies can still apply in how a laboratory chooses to apply a specific piece of evidence and how much weight they give it. One of the aims of the Clinical Genome Resource, or ClinGen, is to work towards having more of a consistent approach to classifying sequence variants. And I won't go into introducing ClinGen much here, as I know other speakers already have. However, within ClinGen, we have this working group called the Sequence Variant Interpretation Working Group. So while the different expert panels are taking the ACMG guidelines and adding in gene and disease specifications, the Sequence Variant Interpretation Working Group makes sure that they are taking a harmonized approach and are consistent in the way that they're specifying the guidelines. This working group also is tasked with developing general recommendations for using the guidelines to help improve consistency. So I'm showing here again the grid from the ACMG AMP guidelines, but I've added in a few additional annotations, such as showing which criteria require gene and disease specifications, and then which criteria have a recommendation from SVI, and which criteria are currently undergoing recommendations by SVI. And you can see there is some overlap in criteria that we've published a recommendation, but it still requires gene disease specification. For example, this PVS1 rule for loss-of-function variants, we published a recommendation of how to determine if your variant has a loss-of-function impact. However, you still have to incorporate gene disease information to understand if loss-of-function is a mechanism of disease. And so that's why some of these criteria you'll see have 2 boxes around them. As far as where to find the different guidance that SVI has put out about using the guidelines, I've shown here a screenshot from our SVI webpage on the clinicalgenome.org website. And here's where you can find general guidance. Additionally, this is where you can find all of the approved variant expert panel specifications to the ACMG guidelines. All 13 of the ones that are currently approved can be found here. So speaking of recommendations, the first one I'm going to talk about is a recommendation we have for modifying the strength of criteria. And this is an important discussion because this will come into play later on when I talk about applying codes. So an important concept to consider is that there are times where the evidence that you're looking at fits with a description within the ACMG guidelines, but the weight is not appropriate, either that it needs to be increased or decreased. So we had worked to come up with a system to be able to identify these instances in which you want to modify the strength of a piece of evidence. What we had decided upon was to, when you're modifying evidence, to use the initial code followed by an underscore and then the new strength. So for example, seen here, segregation data was in the guidelines as a pathogenic supporting piece of evidence, PP1. And if you want to then move it up to a moderate level, you would do PP1 underscore moderate. And if you want to move it up to strong, PP1 underscore strong. And again, this is the nomenclature that the different expert panels use, as well as the nomenclature you would see within our variant curation interface. Okay, so now I'm going to dive into going through some recommendations for using the guidelines. And I've divvied these out into sort of 3 different areas: allele frequency data, variant type and location data, and then patient data. So I'll start with allele frequency data. So there are 3 different codes that involve using population data. We have the BA1, which is a standalone criteria for calling a variant benign based on a high allele frequency. BS1, which is often a criteria used to get a variant to likely benign. And then PM2 if the variant is absent or a very low frequency. So I start with the BA1 code. So this was written in the guidelines as allele frequency greater than 5%. However, from the SVI group, we have suggested a rewording to this criteria to instead read as allele frequency is greater than 5% in any general continental population dataset of at least 2,000 alleles for a gene without gene or variant-specific recommendation. I'll divide this out into kind of the 2 parts we added here. So that first part we added is that this 5% needs to come from a general continental population data set of at least 2,000 alleles. We thought this was important because you don't want to accidentally call a variant benign based on the fact that it was at an allele frequency of 1%, but it was only found in 1 of 100 sequenced people. That you want to make sure your population is large enough that it is adequately representing the true allele frequency of that variant. Additionally, we said that it's okay to use these continental populations. So for example, for this variant seen here on the right, I have a screenshot from Nomad. You can see that the overall aggregate interpretation across all the populations was about 0.9%. However, if you look in the Latino population, this variant is above 5%. So in this case, you could call this variant benign because of this high allele frequency in the Latino population, even though the global population frequency is less than 5%. The other caveat we added to this criteria was that this 5% rule is applicable for a gene that does not have a gene or variant-specific recommendation. We added this because we recognize that for many disorders, a 5% allele frequency is orders of magnitude higher than necessary. Additionally, we wanted to call out the fact that there are non-benign alleles that can be found at a greater than 5% frequency in populations. So for this exception list of non-benign alleles, you can find the list on our website. This was from a publication we worked on for BA1, linked here. And then here's the display of what those variants are that you may not want to accidentally exclude as benign based on some evidence of pathogenicity. And if you think we're missing any variant here, you can also nominate a variant to be on this exception list. So now looking at this criteria again, the So we said you can use 5% unless you have a gene-specific recommendation. So the question becomes, how do you determine what an appropriate gene-specific threshold is? We've been recommending that groups use this allele frequency calculator. I have the URL here. It's from a publication from Within in 2017. And the equation is using prevalence times heterogeneity over penetrance. And this will give you your maximum credible population frequency for disease. And you could then compare this calculated number to the allele frequency to determine if it's high enough to say it is benign or likely benign. Here's a screenshot of what the app looks like, where it allows you to input these parameters, prevalence, heterogeneity, penetrance, to be able to see what your calculated maximum credible population allele frequency is. The way that we have been encouraging some of the groups to use these codes is to here. What we, again, have encouraged groups to do is to try to track the most common estimates, least common estimates, look at what the top gene contributors are, looking at estimates of penetrance. And then when they're trying to calculate this benign threshold, this BA1, we encourage that they use the most conservative estimates. So again, the most common estimate for the prevalence of this disease, the top gene contributor, and then a lower estimate of penetrance. So in this case, putting in these parameters for hypertrophic cardiomyopathy would give you a allele frequency of about 0.1% threshold to be able to call a variant benign. So when it then comes to actually now looking at the population databases, a lot of the groups are using primarily Nomad to look at this information. I just wanted to call out that within the variant curation interface, you will see both Exact and Nomad displayed. However, Exact is incorporated into Nomad, and so you don't want to aggregate across these two groups, but rather use one or the other when you are determining if your variant allele frequency is met. Additionally, when looking in Nomad, we've been encouraging the different expert panels to use the filtering allele frequency. So what this is you'll see the filtering allele frequency is displayed sort of in this top section, this pop max filtering AF. And so what this is the filtering allele frequency functions as an equivalent to the lower bound estimate of the true allele frequency. So it builds in a little bit of confidence to what the calculated allele frequency is. So for example, here you can see that in the African population is where you saw the highest allele frequency for this variant at about 0.19%. Then you'll see the pop max filtering allele frequency is at about 0.14%. And so this pop max filtering allele frequency will always be lower than the calculated one because it is, again, it's building in this confidence interval to indicate what potentially the true allele frequency of this variant could be in the larger population. So it is a conservative way of looking at what the true allele frequency for a variant could be. And so we recommend is once you've calculated what your BA1 or BS1 threshold is to then compare it for any given variant to that variant's filtering allele frequency to determine if this criteria is met or not. So now looking on the pathogenic side, it's going to talk about applying this criteria for absence. This PM2, it's a pathogenic moderate piece of evidence. So it's written as absent from controls. However, a question that often comes up when looking at dominant disorders is, does it truly need to be absent to be able to apply this criteria? Looking at the frequently asked questions page from Exact and Nomad, you can see that this organization has made an effort to exclude individuals with severe pediatric diseases, and so they don't expect the dataset to be enriched for these individuals. Additionally, at least for Exact, the age range was primarily older. About 70% of individuals are above the age of 40. And these are important considerations for when you're looking at a disease that may have later onset, that you may not expect that disease or variant to truly be absent from Nomad because it would have presented later in life. So a conservative way to look at Exact and Nomad is to not view them as controls, but instead more of the general population, and to be able to compare your estimates using that approach. So I've shown here how the different expert panels have specified PM2, where you can see that for some of the more severe diseases, they've had a much more conservative number for what they call absent, whereas some of the more common diseases and those that are recessive, allowing for a higher frequency to be able to say that it is fits in this absent or rare range. However, one general recommendation that has since come from the Sequence Variant Interpretation Working Group is a recommendation to only use this absence or rarity information as a supporting piece of evidence towards pathogenicity instead of a moderate piece of evidence. And that's because we think that rarity on its own is overweighted in the current framework. Okay, so next I'm going to move on to variant type and location information. We'll start with the pathogenic very strong criteria, PVS1. This criteria is for null variants in a gene where loss of function is a known mechanism of disease, and the guidelines include these additional caveats that you need to consider. So you need to consider, for this gene, do loss of function variants actually cause disease? But then also consider, does my variant actually have a loss of function impact? And so it's sort of two different questions to address. And a few years ago, the SVI group published guidance on interpreting this PVS1 criteria that I've shown here, that you can find from our website. And again, one of the first questions that you need to address is loss of function a mechanism of disease? And there's a couple ways you could figure out this information. We have some suggestions here taken from the paper. One way that I often will approach looking if loss of function is a mechanism is to look at the ClinGen dosage sensitivity curations. And these actually can be found on the ClinGen website. So I've shown here, I'm on the home page for clinicalgenome.org. And in this case, I'm going to use MYBPC3 as an example. So I typed this into the gene search box. And this takes me to the gene curation, the landing page for this gene on the ClinGen site. I can see this gene is an established gene for hypertrophic cardiomyopathy. Additionally, it has gone through this dosage sensitivity curation, and it was scored as a 3, which is the highest score for showing that haploinsufficiency can cause disease. However, this curation is not available for all genes yet, so there could be times you need to look elsewhere to determine if loss of function is a disease mechanism. One way is to look at population databases to be able to determine if this criteria could be met. So Nomad and Exact calculates this PLI score, which is the probability that a given gene is extremely intolerant of loss of function. The score is between 0 and 1, with scores above 0.9 suggesting extreme loss of function intolerance. And there's also this observed expected ratio that looks at how many expected loss of functions there would be versus observed. However, it's important to consider that both of these are predictors. These are not based on manual curation of if loss of function is intolerant or not. These scores are also mostly only applicable for dominant disorders, but are also really only applicable for diseases that would emerge post-reproductive age. Again, because NOMAD makes an effort to exclude severe pediatric onset diseases, but not adult onset. So if your disease would not impact reproductive health, you would not expect to see an absence of these types of variations within the population databases. So again, looking at MYBPC3, which we had shown loss of function is a mechanism of disease by looking at that dosage curation, in NOMAD, you can see that the PLI score is 0, showing that it is not intolerant. Again, because hypertrophic cardiomyopathy is later onset, and so you would not expect to see an absence. Whereas a gene like ZEB2, which has a severe pediatric disease, you can see the PLI score is 1, because in this case, loss of function does cause disease, and that disease is earlier onset. All right, so now that we've addressed whether or not loss of function could be a mechanism of disease, we now need to interpret whether or not our variant actually has a loss of function impact. From this publication that the SVI had worked on, we worked through this decision tree for different variant types of considerations that you need to look at to determine if loss of function is an impact. I've shown here the nonsense and frameshift parts of the tree, and then it continues on to looking at deletions, duplications, and initiation codons. So I have one example that we can work through. This is a frameshift variant in MYBPC3. And so the first question we have to address is whether or not this variant is predicted to undergo NMD or not, nonsense-mediated decay. So shown here is a screenshot of from Allele of looking at where in the gene this variant occurs. So the actual cDNA change occurs within exon 30 here, but since it's a frameshift variant, I want to look at where the actual stop codon occurs, which is in the next exon in exon 32. However, there are 35 exons in this gene, so this stop codon still is occurring before that boundary for NMD. So I can say that in this case, I believe that this variant is predicted to undergo nonsense-mediated decay because it's before the penultimate exon. So the next question I need to address is this exon present in biologically relevant transcripts? This is another case where looking at the Nomad data can be helpful. So I'm here on the MYBPC3 landing page, and I'm showing the different transcripts, and you can also look at the expression of these different transcripts. Our variant is present here where the arrow is pointing, and if I follow this variant up, I can look at which transcripts it's in, and I can see it is present in the canonical transcript, this top transcript, which also has the highest expression levels. So in this case, I could conclude that my exon is in an exon that is present in the biologically relevant transcript, and now feel confident applying PVS1 at the highest strength. All right, now I'm going to look at some of the criteria that's applicable for missense variants, specifically looking at PP2, which is a supporting pathogenic code for genes in which missense is a common cause of disease, and PM1, which is a pathogenic moderate piece of evidence for if the variant occurs in a mutational hotspot or well-established functional domain. So the question often becomes, how do you determine if these two criteria are applicable or not? And for both of these, being able to use population databases can also help determine if these are criteria are applicable. So looking at the PP2 code, the one from Missense Constraint, it's helpful to look at the Missense Constraint score that is calculated by Nomad, looking at the number of observed versus expected missense variants. The exact paper that had come out suggested that any gene with a missense constraint score above 3.09 suggests significant missense constraint. So seen here for the MYH7 gene, you can see that the missense constraint is above this, so in this case, I could apply PP2 for this gene. However, you often sometimes want to look at regional constraint, and this is helpful when you want to find specific domains that are more highly constrained as opposed to the entire gene. So right now, in order to look at regional constraint, you have to change the data set up there in that top right corner to EXAC. I believe this is soon coming for the Nomad data set, but right now it's only been calculated with the EXAC data. So if you go to the top and change to EXAC, you'll then see this bottom section appear where you have this regional missense constraint bar. In this case, it's on a score of 0 to 1, in which case missense constraint scores closer to 0 indicate this region is more intolerant against missense variation. So for this gene, you can see that the first part of the gene, as this gene is read 3' to 5', so this yellow part here has a more constraint for missense variation than the rest of the gene. And when you kind of compare down below, when you look at where the ClinVar likely path variants are, you can see there's more clustered on that right-hand side. And if you look at the frequency of the variants then below it, you can see also there's more high-frequency variants on the left side. So again, both of these, looking at the population databases can help determine whether or not these criteria are met. However, it comes with the same caveats as the loss of function scores, in that you really would only expect to see constraint if this is in a gene where the disease can impact reproductive health. Otherwise, you wouldn't expect to see a constraint within Nomad. Additionally, right now, these are also more applicable for dominant diseases because you would see that constraint as opposed to recessive diseases. All right, so next I was going to look at now patient data. So another question that often comes up is when you have multiple people who have disease that have had this variant, how do you incorporate all of that information and what criteria is applicable? So you can see I've highlighted some of the more patient-centric pieces of evidence here in the framework. One important consideration is that when you're looking at case-level data, you want to gather all of the evidence available and not limit it to the current case in front of you. So I have a screenshot here from the ACMG publication where they specifically addressed this and said that you should look at all cases. So for example, if you want to apply the de novo criteria, it doesn't need to be met by the current case in front of you. If you've seen this variant before and it was de novo, or even in the publication it was de novo, you could apply the de novo criteria based on those other observations. It doesn't have to be limited to the current case in front of you. So speaking of de novo criteria, there are two criteria in the guidelines with regards to de novo occurrences, a criteria for when it is an assumed de novo versus confirmed. And the assumed versus confirmed here is with regards to if you are confirming paternity or not. So in both cases, you are confirming that the parents don't have the variant. The difference between the strong and moderate codes are whether or not you then took the next step and confirmed the parents are the parents. And another important consideration when applying the de novo codes is looking at the patient phenotype and seeing if it actually is a good match with the disease. Any given individual has a handful of de novo variants, and so you want to make sure that it's not just by chance you found a de novo variant in this gene, and that actually that the phenotype you're seeing in the patient would be associated with variants in this gene. So SVI published guidance about how to apply these de novo rules, and I have shown here where you can find it on our website. We wanted to publish guidance for how to use these de novo occurrences because a question often comes up of if when you have multiple de novo occurrences, how do you add these all together to figure out the applicable strength? So what we had done is in this top part of the table, sort of separated out de novo occurrences into two different scales. One, whether or not the de novo occurrence was confirmed versus assumed, and then also looking at phenotype specificity, if the phenotype's highly specific, consistent, or consistent but highly genetic heterogeneity, and then kind of divvy out the points from there. So you can go through and gather all the points applicable for all the probands you've seen, and then look at that bottom table to see what the sum is and determine what the applicable strength should be. So I have an example here. Let's say you have a de novo variant in NIPBL, which is associated with Cornelia de Lange syndrome, and you've seen it in 3 probands. This is a specific phenotype, so you could use that top bar, the phenotype highly specific. Let's say you had 1 confirmed de novo occurrence, that would be 2 points, and then 2 assumed de novo occurrences, that is 1 point for each one of those. So in the end, you would have 4 points, and if you look at that bottom table, you can see it's very strong. So in this case, you can modify the weight of your de novo occurrences up to a very strong strength level. Another question that often comes up is looking at recessive diseases and how to modify the strength of those. SVI has also been working on guidance for SVI, and one thing I wanted to call out is we also have put in some slight rewordings to this criteria. We added in that for these recessive disorders, that variant detected in trans could be pathogenic or likely pathogenic. Additionally, these observations should be in an affected patient. The description of these in the ACMG guidelines did not specifically say the person needs to be affected, but we think that is an important consideration when you're actually applying these this criteria. And again, a question has often come up before of when you have multiple observations, do you upgrade, and would you even want to downgrade in cases where you are unable to confirm the phasing? So similar to that de novo framework, we've come up with a points-based approach to determine the applicable strength of all of your recessive observations. So in these cases, the points are divvied out according to if the variants are confirmed in trans versus phase unknown, and then also separated out based on the classification or zygosity, with less points for homozygous occurrences or uncertain significance variants on the other allele. So I have an example here. So let's say you have found this variant in 5 probands, 2 of which were homozygous occurrences. Those would be 0.5 points each for 1 point total. And then you've also found this variant in 3 probands who had a confirmed pathogenic variant in trans. That's 1 point for each of those for 3 points total. If you then sum across all these observations, you have 4 points, which can then move up again to a very strong strength level. So while the PM3 code is applicable for being able to case count recessive observations, a question often comes up of how do you then count for multiple probands who have an autosomal dominant phenotype. What is the criteria you would use for those? So in the guidelines, there's this PS4 rule for the prevalence of the variant in affected individuals is significantly increased compared to prevalence in controls. So this criteria is often, the way it's written, more applicable for case control studies. However, often you don't have enough data to really perform a statistically significant case control study. And so in the guidelines, they address this by adding this note 2 that I've circled below, where when you have a very rare variant where a case control study may not be significant, the fact that you've seen this variant in multiple unrelated probands and that this variant's absent from controls, you could then use this code for these observations as well. So from SVI, we have suggested that another use of PS4 besides just these case control studies is you could use this when you have multiple unrelated probands with consistent phenotypes. Again, especially for autosomal dominant diseases, as the recessive diseases would be used under the PM3 rule. And here's just an example of how some of the different expert panels have specified this PS4 rule. You can see that for diseases that are not as phenotype specific, like hearing loss and cardiomyopathy, they require more probands at each strength level compared to some of the more specific phenotypes like Noonan, in which there are less probands needed for each different strength level. So another criteria to discuss is this PP4 rule, pathogenic supporting piece of evidence, for when you have a patient's phenotype or family history that is highly specific for the gene. From SVI, we have thought that this criteria could also be used when you have some non-genetic confirmation testing. So for example, for PAH, if this individual had gone through biochemistry testing and shown to have high phenylalanine levels, you could then apply PP4 for that observation, because even though it is a sort of a clinical test, it's non-genetic, so it's not variant specific, but rather is more confirming that this patient really does have this specific phenotype. Same with our mitochondrial expert panels, who have used this code for observations of reduced electron transport chain activity in patient cells. Again, this is not a variant-specific functional assay, but rather is just confirming that your patient has this more specific phenotype. And so this is sort of another way in which the PP4 criteria could be used. So again, taking a step back, looking at all of the different proband codes that we have here and how you would use these for different observations. We suggest that you would use the PS4 spectrum when you want to count probands, especially those for dominant disorders. PM3 is the spectrum where you would use when you want to count probands from a recessive disease. Additionally, de novo observations can be counted under this PM6 or PS2 spectrum. And if you then have a proband who meets this very specific phenotype code, you could then also be applying this PP4 rule. All right, so lastly, I wanted to get into a bit of what we've been working on now with regards to how to combine different criteria to reach a classification. In 2018, Sean Tabat-Tijan from the SVI working group published this approach of using more of a Bayesian approach to the ACMG guidelines. What he did was look at the thresholds for each classification level, so pathogenic having a probability of disease of 99%, likely pathogenic at 90%, and then looked at the combining rules in the framework and was able then to back calculate what is the odds of pathogenicity at each strength level. So for example, a pathogenic supporting piece of evidence came out to be at about 2-to-1 odds of pathogenicity, moderate at about 4-to-1 odds. And so what this means is this is now a helpful way that when you want to calibrate your specifications for a gene, to be able to use these odds to determine what is the right strength of evidence. So for example, how I mentioned before, we have suggested that absence, PM2, should be moved down to supporting. And that's because a moderate piece of evidence should be 4-to-1 odds of pathogenicity, meaning for every 5 rare variants, 4 of those should end up as pathogenic. We don't think that that is an appropriate estimate, and so we have suggested moving it down to supporting. You could also use these odd levels when you want to, again, calibrate maybe a in silico tool like Ravel. If you want to determine at what threshold is applicable to say for this gene, a score would in a certain range suggest pathogenicity. You can then calibrate knowing that at a supporting strength level, you need about 2-to-1 odds of pathogenicity. So while this odds approach is helpful when you want to determine the applicable strength, when it comes time to then combining all of your evidence types, it's helpful to move now to this compatible points system. So where it's Bayesian in the back, but now points when it comes time to combining rules. So I've shown here now what is the point value at each strength level, and then the sum total levels that would get you to each classification level. So this would be helpful in using this point-based approach for multiple reasons. One would be, this would be also helpful, this point approach, for using criteria combinations that are not specifically listed in the 2015 guidelines. For example, if you have a loss of function variant, PVS1, plus absence from controls if you've moved PM2 down to supporting, this combination, very strong and supporting, is not in the guidelines, but this sums to 9 points, which is in the likely pathogenic realm. Additionally, using the points could help us differentiate VUS. Because VUS is such a wide category, you aren't able to adequately differentiate those that are more suggestive of pathogenic versus those that are more suggestive of being benign. So for example, if you have a variant where all you had was an in silico prediction, that's VUS, but only with 1 point total. Let's say you also have a variant where the in silico tools suggest damaging. It's also in a critical functional domain and was found in trans with a pathogenic variant. That is also an uncertain significance variant, but at a 5-point range, so really close to that boundary to likely pathogenic. So being able to take this approach, we can now better divvy out those VUSs that are more suggestive of pathogenicity and figure out if it is more appropriate to return variants in that range versus those that are uncertain significance and closer to the benign side. So in conclusion, I just want to acknowledge the ClinGen sequence variant interpretation working group, especially my co-chair, Les Bieseeker, and also thank all the different expert panels that are working on the ACMG AMP guidelines, because being able to get all these different use cases from the expert panels is really helpful in being able to inform our process and work towards more consistent classifications across laboratories. Thank you.